Hello. So I guess we're getting started now. Am I right, Megan? All right. Today, um, we're here to present to you about the Dragonfly Mercury project. This project is a nationwide study that is being conducted by the University of Maine, USGS, and it's being hosted by the National Park Service. And we are a citizens group called Citizens United to protect the Maurice River and its tributaries. And we have been conducting the collection of samples here on the wild and scenic Maurice River. And so CU Maurice River is dedicated to protecting the watershed of the Maurice River and the region known as Down Jersey, thereby enabling current and future generations to enjoy environmental, recreational, cultural, scenic resources of this wild and scenic global treasure. We were founded in 1989, and what we do is we try to engage the community with volunteer opportunities to protect our area. In 1993, the Maurice River was designated as wild and scenic, and it is part of the National Park Service. And that is how we came to get involved in the Dragonfly Mercury project. So here tonight with us, or this afternoon with us, we have me, Carla Rossini. I'm the program manager here at CU Maurice River. And also we have Megan Thompson, who's the assistant office manager. Now, I would like to stress that we are not scientists, and the information that I have here, we have not compiled. What we've done is we have gotten people engaged, volunteers engaged with the Citizen Science Project, and we've done the data collection, which the samples were then sent to the University of Maine for analysis and on a nationwide level. So mercury distribution. How does mercury get into our ecosystems? Well, the EPA says that the source of mercury is actually anthropogenic. When we burn coal for energy, it's emitted up into the atmosphere. The mercury is emitted up into the atmosphere. The wind currents carry it. And then with precipitation, whether that's rain, snow, fogs, clouds, that mercury settles down and covers everything. You have mercury on you right now. The trees have mercury. All the landscape has mercury. And what happens is that also when it rains, the runoff will carry that mercury into streams and then also into large bodies of water like lakes, bays, oceans. As that mercury settles down into the water, organisms will consume it, like the small invertebrates or macroinvertebrates like dragonflies and damselflies. Now, dragonflies specifically are pretty high on the food chain, and so they accumulate a lot of it. They bioaccumulate it into their system. And this is what makes them a good indicator species as to the health of the ecosystem. And it shows there by analyzing them, we can see how much mercury is actually in the ecosystem and how that's going to affect the rest of the animals higher on the food chain. So in this study, that is a national scale assessment of mercury bioaccumulation in the United States national parks using dragonfly larvae as biosentinels through citizen science framework. This was done by scientists at the University of Maine. And the goals of this program was, one, to implement a national mercury monitoring project in protected lands, so in national parks, and two, to assess geographic variation of dragonfly mercury concentrations across habitat types. And finally, to establish dragonflies as an effective biosentinel to inform potential exposure risk at continental scale. So a lot of the information, what I would like to do here today is, again, I'm not a scientist, so what I would like to do is to tell the story of our citizen scientists who have gathered the specimen and for the University of Maine to analyze. And but the information, the graphs, all of the analysis of data that you're going to see in this presentation all comes from this report. So I really recommend going online, putting this report in. You can even do a screenshot right now to make sure that you get the title right. It is a longer title. And you can go on to the USGS. A lot of these maps that I'm showing are actually interactive. And so maybe you're in New York, or maybe you're in Pennsylvania or Maryland, and maybe there's a park that's closer to you that's done a very similar study or has submitted their samples to the University of Maine, and you can see how your area is faring when it comes to mercury deposition. So, a question: why dragonflies? Why would we want to collect dragonflies for analysis and not other invertebrates? Why not birds? Why not fish? Well, dragonflies are obligate predators by nature. They feed on a lot of different insects in the water, aquatic insects in the water. They can feed on small tadpoles, small fish. They're really a consume-all species, which means that they also bioaccumulate a lot of that mercury, being higher, as we said before, higher on the food chain. They inhabit a large variety of freshwater habitats. They're in lakes, they're in ponds and streams and large rivers, they're in wetlands. And so that means that we can find them in a larger range in the United States. And they have narrow trophic range. So they're going to, they're not going to travel very far. They're going to always come back to the same spot. They're not going to be taking, like a bird, they're not going to be feeding in a pond and then later feeding in a stream. They're going to be staying in one site. They're not going to be going too far away from their feeding grounds. And mercury is also present in their tissue, and a lot of their tissue is methylmercury. And then there's high site fidelity in dragonflies. So, and then another reason, we have to look at the utility for the study. So in this paper, they say that another reason why they decided to use dragonflies is because they needed a species with the characteristics that we just spoke about, but also one that could be studied on a continental landscape, right? So this study was proposing to look at the mercury distribution throughout the entire United States. So what species or what animal could we use to study mercury deposition and levels in the United States? So there's a nexus to human exposure in that the level of mercury in the dragonflies correlates very well to game fish. So since humans obviously consume game fish, then and those levels mirror each other, then that creates a nexus between the dragonfly, the level of mercury in the dragonfly, and the how a human would be affected by it. Dragonflies are widespread throughout the study area. They're in all 6 continents. They occur in a variety of habitat types. As we said before, they have that high site fidelity. And then also there aren't as many logistical, ethical, or regulatory limitations as there would be if we were collecting fish, if we were collecting birds, if we were collecting salamanders, frogs. And then also, now I think my volunteers might disagree with this one, especially after the last site we did, but they say that there's an ease of collection and identification. So if you speak with a first-year volunteer, they might say that it's not so easy to identify them, but you can, after the second season, you get an eye for identifying and collection. Some days we collect quickly, some days it takes us a little bit longer. So what dragonflies? So there's 6 families. So we get the gumfoot here in our area. We've collected gumfooti, which are the club tails. We've collected the Aishnidae, which is actually the one that we collect most. And I believe in the study, they said that 41% of the samples that are sent in are Aishnidae. You can identify them by the very elongated body that they have and these very big bulbous eyes that are set out to the sides of their face. The Catalegastridae, which are the spike tails. These, for me, it looks like they almost have hair on them. They're burrowers, and so when you pull them out of the water, it looks like they have like almost like the soil or the sand stuck in little hairs, and that's always the easiest way. But another way is that they have this really zigzag jaw on them. The Cordelidae, or the emeralds and green-eyed skippers. They have a very wide body with 4 points at the bottom of their abdomen, right around their anal cone. The Libellulae, this is one that we send in a good amount as well. Those are the skimmers. And for me, you'll see a difference between the Cordelidae and the Libellulae, because the Libellulae, their eyes are also more bulbous than the Cordelidae, which is a similar, has a similar anatomy. And the Macromidae, this one looks, when you pull it out of the water, it looks like a spider that's missing 2 legs. I mean, it's very, very spider-like. The ones that we've pulled out have been actually quite large, and I would say that this is the least frequent one. So we've pulled the least out of these. And so nymphs of a family look similar, while the adult species vary greatly. It's almost easier to identify the adults than the immature species. So how did the program get started? So the program got started actually with a student a school-age student who had a science project to do, and they decided that they were going to collect dragonflies and study mercury contamination in a local waterway of theirs. And so they did that, and the data that they pulled out was really quite interesting. And so their science teacher said, well, why don't we send this to the University of Maine? So they sent it to the University of Maine. The University of Maine said, oh, wait, this is a great opportunity to study, all across the United States. And so I thought, well, how could we do this type of study, from New Jersey to California and Minnesota to Florida? How could we do that? And they decided that a great partner would be the National Park Service, because the National Park Service is all about engagement and creating appreciation and awareness. So, and they also have preserved lands all throughout the United States. And so they decided that they're going to collect the dragonfly species. And then, so how were they going to collect the dragonfly species? Well, through citizen scientists. So they put together protocols and started training leaders like CU Maurice River on how to do a collection without spoiling the samples. And the reason why CU Maurice River is able to participate in this is because we're what's called a partnership organization to the National Park Service. So the National Park Service has programs under it, one of it being the Wild and Scenic. And the river that we work to protect and also to engage the public with is a Wild and Scenic river, a unit of the National Park Service. So our National Park Service river manager, Paul Kenny, contacted us and asked us if we would like to engage our audience with this or get our audience involved with this study. And we said, absolutely. And so that's how we got that's how we got the audience, the public involved. So we gathered together the troops and we went out to the streams with our dip nets, and they all started collecting the samples. We would bag the samples, tag the samples, and send them into the University of Maine. At the University of Maine, they would dry out the samples and then weigh the mercury content of each one. And then they would file and analyze that data as it came in throughout the entire United States in order to better understand what the rates or levels of mercury are in different ecosystems. Excuse me, in different regions. So the data collected, there have been 9 years of collecting from 2009 to 2018. Over 100 national parks have participated, and each park does between 2 to, I believe, 6 sites. So there have been 450 different sites that have done within these national parks. And all the sampling has been happening in the 5 different habitats, which is rivers, streams, lakes, ponds, and wetlands. And over 4,000 citizen scientists have participated in this program. 15 dragonfly larvae are collected at each site, which means each park is collecting 45 samples each year. So 14,831 samples have been collected. When they go into analysis, there are intrinsic and also extrinsic factors that they're looking at. So we try to collect samples that are at least 12 millimeters long. And the longest one I think we sent in was just shy of 40 millimeters. I mean, it was an Aishna that was huge. Oh my gosh, I remember when we pulled it out. It had just come out of its exoskeleton. We were all amazed by it. And then also the taxonomy. And extrinsic factors that can affect the amount of mercury that are in a specimen are habitat type and the ecoregion as well. The graph that you see on the right is showing the means at the different national park units. You can see a long list there. Now, this graph is only showing data from 2009 to 2018, but the study is continuing. We actually just finished 2020, I believe, last week, our sampling for 2020, but that information isn't included here yet. All right. Oh, and then in the bottom right-hand corner, you can see a map of the United States, and you can see, including Alaska and Hawaii, and you can see all of the different sampling sites. So what to expect? This is a slide that I show our volunteers. We have the draggers, the baggers, and the taggers, right? The draggers are the people who go in the water. They have their life jacket on, and what they're doing is they're searching for the samples. They go around and they're looking for the specimen, they're collecting them. They hand them off to people with what we're calling cleaner hands, not clean hands, cleaner hands. And those people will take them back to, those are also our safety people. They're keeping the eyes on the draggers. They're making sure that everybody's safe. And they're carrying and shuttling the specimen from the draggers to the taggers. And we ask them always to be as clean as possible. Now, the taggers, these are the detail-oriented volunteers. They're the identifiers, and they will have their gloves on, and they're using little plastic spoons. They're not allowed, they can't touch the specimen because we all have mercury on us, on our hands, everywhere. If we touch that specimen, once it's been rinsed off in clean water, then we're actually contaminating it, and we could be putting more mercury on it than what it would have just coming out of the water. And so the gloves are 100% necessary. What they do is they ID from cards that we have, and then they measure the specimen. They put it in one bag. They fill out a tag. So basically giving the specimen a number, and then they double bag it. They put the little bag with the specimen in another bag, and they put a little piece of paper with all of the information on the site in the park and the like in it. and we try and make sure that they are as sterile as possible. So that's kind of a little overview of what we're doing when we're out in the field. So we spoke about different factors that can contribute to different mercury quantities being in a sample, and one of those things are their habits. Are they claspers? Do they cling to flora or emergent flora, or are they burrowers? Or are they underneath the ground, underneath that riverbed and burrowing underground and waiting for the prey to come by to grasp them? Are they sprawlers? Do they sit on top of fallen logs in the water and they're waiting as something, a prey floats by? Or are they hiders? Do they tend to be in the leaf litter and detritus underneath in the water? So, what are their habits, and how does that expose them? Knowing this also helps you find them in the water as well. So those variation levels, so that we can see that the species and their habits affect it. Some species like the Aishnadai here, you can see that they can live anywhere from half a year to 6 years in the water before they emerge as adults. The Libellulae, for example, is a third of a year to 3 years. So on average, the Aishnadai can spend 2.6 years in the water, whereas the Libellulae will only spend on average half a year in the water. So one is exposed more to mercury in its immature form than another species is. So this makes sense. And then, also their habits, their hunting habits. And in the graph just below, you can see that the Aishnadai has a higher concentration of mercury in it, or on average has a higher concentration of mercury than the Gumphidae or the Libellulae, which is the Libellulae is the third column there. Another varying factor is the habitats. So you'll see that rivers and streams, rivers have the highest quantity of mercury, then streams, then wetlands, lakes, and ponds. But there are other factors that play into this variation as well. For example, is it an intermittent stream or is it a perennial stream? Is it a stream that's mostly surrounded by wetlands or not? So all of these factors can play into the amount of contamination that are in those habitats. So sample sites on the Maurice River. Here you can see we have 4 green stars and an orange star here. So, there are 5 green stars and 1 orange star. So those are the sites that we've done from 2015 to 2020. And you can see delineated in that dark blue line there, that is the wild and scenic designated area. So that's the main stem Maurice, and the river, the northernmost river, that is the Manamuskin, that is the Menantico. The river there in the middle, the tributary in the middle is the Manamuskin, and the shortest little one down there is the Muskie. And here I've included some shots of the different sites and some of the volunteers who have participated. We have our regular volunteer base who really enjoy the camaraderie of getting outside and doing good for the environment and for community health. And then we also have school groups that come out with us. Here in these pictures, we had a local high school who joined us for the sampling. And this is at the Maurice River site in Vineland. You can see here on the right, that is a former watershed ambassador, and she is working one of the outstandingly remarkable resources of the Maurice River, which is the stands of wild rice. That's a tiny little one, but we're pretty far upstream here in this picture. And on the left, you can see that this is a public beach area. And in the center, you can see the baggers trying to fish out the specimen that like to hide in the leaf litter. This is the Manu-Muskin River. This is a harder, this site is actually considered to be a pond that is surrounded by wetlands. And you can see here the volunteers are standing in a patch of lilies, and it's really a gorgeous site. Mobility is a little hard, so we tend to use the canoes to do sampling, so we can get out and about a bit easier. These are some of the samples that we pulled out, and Cheryl Howell is our volunteer guru. She has been involved since the very beginning. She does all the site checks with me, and if you can't find a bug, you got to talk to Cheryl, because she'll tell you where they're hiding that day. She can probably even tell you, based upon the temperature and the light and the tide, exactly where they're going to be. That's our Cheryl Howell. She is a phenomenal volunteer at the Dragonfly Mercury project for Citizens United. So they're the specimen that we're looking at. Those, it looks like we have 1, 2, 3, 4, 5, 6, 7 Cordyligastриdae and 1 Aishnidae. And if you look at those, the specimen that are quite a bit larger, you can see what I was talking about. That it looks like they have little hairs on them, and that the and that the sediment gets stuck in it. And that's what I was referring to before. And the Aishnidae is in the top right-hand corner of that ice cube tray. And you can see on that one, you can see its big bulbous eyes and the elongated body that comes into a pointy anal column. So that looks like it was a successful day, and we have some really nice big specimen there. And Cheryl, the other thing, Cheryl is dressed to perfection here. She has every tool you need. She has she has her dip net there. She has her life jacket on. She's got a bucket that we fill with water, that one that's just laying on the top of the water that's connected to her life jacket. We fill that with water, we put the specimen in there, and then as the baggers come, she will pour that into the little yellow tray that she has so that the baggers can shuttle it back and forth. So that's actually a good picture of what we look like when we go into the water. And here, this is another site of the Manu-Muskin. This is actually the stream that goes into the pond. And one of the varying factors can also be the tree cover. So here we're looking at maybe, I think we're about 75% tree cover here, which will give a bit of protection to the water. This is the Menantico River. And this is quite different. So it's quite open, but it's a small stream, has little wetlands, little pockets of wetlands on the way down. As you go downstream, the bed is going to turn from sand to gravel. And our volunteers really love this site because of the diversity of animals that we pull out. We have Joseph up top showing us an eel, and in the small picture at the bottom we see it just escaped me, a stonefly. So a group of volunteers standing around the table after we had already finished bagging and tagging. These are some more pictures. These are, you can see on the left, the stream that flows into the pond, and you can see some of the wetland areas there. This is the muskie. We've actually, in the study that we're talking about, the muskie only has 15 samples that were analyzed for that. It was a new site that we added on, but since we've done it 3 times and we'll be moving on to a new site, should we be included in this study again? On the left, you have our river manager from the National Park Service. He likes to come out and support us, and he's been really integral in keeping us involved in this study and making sure that we have what we need. And he's really been a great advocate to make sure that the study continues on the Maurice River. And again, you can see us, sometimes it's easier to move about in canoes versus on foot. And this is one of our, another favorite place for the flora lovers. The wetlands that surround the Muskie are just, the diversity in flora just makes it an absolute pleasure to go out there to collect the dragonfly species. And we have volunteers of all ages that join us. Sometimes at this site, it is it's affected by tide, so we have to really plan when to go out there, because sometimes if you get the tide wrong, we won't find any specimen. But then, you know, we have to go out at a very low tide. It has to be almost a warm day, it seems. That's what we're putting together. But when we do collect there, it seems like we always get the biggest, fattest specimens that you can find. So the habitat type. So looking at the paper that was just published, that we mentioned before, they have denoted the Maurice, the main stem Maurice. They're saying that it's a river, and it definitely has the peripheral wetlands. So you can see that those, with those characteristics, it's going to have a higher amount of mercury. The Menantico, they have defined as a stream, and in areas where we studied, I would definitely say that it has the, in some areas it's a perennial stream, in other areas it does have the peripheral wetlands. The Manumuskin, they've said, is a pond, which actually holds, if you look at the pond levels over here, the pond levels are actually smaller, or lower, excuse me, than lakes and streams. So, and that actually holds true with the results that they've sent us, that at the Manumuskin, the Cumberland Pond site, is where we have our lowest numbers. And then the Muskie is also being defined by them as a stream. So the data from the Maurice River. From the over 100 sites that have been tested, it seems that the Maurice River is the 4th highest in the United States out of the sites that have been tested. Remember that these are all national parks. So in this study, the results have come from the 2015 to 2018 samples that we sent in, where we sampled at 5 sites. We had 12 different sampling states, 12 sampling dates, and we sent in over 180 samples. And involvement, in those 3 years, 152 volunteers and 736 volunteer hours were dedicated to this project. And in the bottom there, you can see in that little bar a cutout. And the bar, if you look at the Maurice River, you'll see that there's a bar that's the mean mercury for the entire park. And then the circles are the mean for each site. And with that, I would like to introduce Megan, who is our assistant program manager. Excuse me, is our assistant office manager.
Hello, everyone. Thank you for joining us today. And I just wanted to say, if you enjoyed this presentation and would like to become involved, we want to invite you to join CU Maurice River as a member. By joining CU Maurice River, you'll get to meet others who share a love for the great outdoors and want to make a difference for their environmental and community health. As a member, you help us reach our mission. Doing this Dragonfly project is one of those ways in which we attempt to reach our mission, which is to help protect this river that we love so very much. Here we have over 1,200 members, and they donate their time, their energy, and their funds to help keep us going and help us to support our programs. If you decide to join us, you get to enjoy the camaraderie of other green-minded members and join a team of volunteer stewards working to protect the natural and cultural resources for future generations. So if you'd like to join us, please feel free to send me an email at megan.thompson@CUMauriceRiver.org.
All right. And with that, I would also like you to, if you are interested in learning more about the Dragonfly Mercury project, I would like to invite you to go to the USGS site or the National Park Service site or the University of Maine site, and you can download that report that we've been speaking about. And the interactive maps and graphs are really, really interesting. So I really invite you to see on those maps what park is closest to where you live, and see what the mercury contamination or the lack of mercury contamination is close to where you live. If you're interested in joining us, we'll be sampling again next year. And my name is Carla Rossini. If you would like to get involved, my email is just carla.rossini@CUMauriceRiver.org, or just info.org or info@CUMauriceRiver.org. Thank you all for joining us, and hopefully we'll see you about on the river.